Great. Thank you, Clive, for that nice introduction. So yes, I've been well, I was trained in pediatrics. I've been involved in clinical trials of TB vaccines for now 20 years, and it was very early in that journey that I had this very modest grant from Immunopaedia, which sadly did not turn me into a card-carrying immunologist, but it did allow a clinical researcher to get into the laboratory and to become immunologically literate, for which I have been very grateful on more than one occasion. So thank you for the invitation to this very inspiring meeting. I'd like to tell you a story. I'd like to tell you a story of what is just over a century of development of vaccines against tuberculosis. And by the time I've finished, I'll hope you get a sense that if we are not quite at the end of that story, then we are perhaps writing the final pages of that final chapter. But to understand why we need a new TB vaccine, it's necessary to go back to the beginning. Now, we work out how to advance our slides here. Here we go. Okay. Now, the earliest evidence, pathological evidence of tuberculosis, can actually be found in the Pleistocene area, the late Stone Age. But we have to wind forward a few thousand years. And here we've got evidence from the necropolis of Thebes in New Kingdom, Egypt. This is about 3,000 years ago, where folk have isolated M. tuberculosis DNA from the spinal tissue of mummies. Now, we don't have lung tissue, but we have evidence, and both pathological evidence of tuberculous osteitis, as well as evidence of MTB DNA. Now, we don't know whether these folk died of tuberculosis or with tuberculosis. But since these are adults, we do know that they lived for a considerable period with disease. And if they had pulmonary involvement, then they most likely transmitted that disease to their children. And this is a key component of understanding the fight against tuberculosis, is that the disease is geared towards transmission to children and other contacts. We do know that historically tuberculosis was a great killer of humans. And here we have data, because we're scientists, we like data. This is data from 1632, from the Bills of Mortality of the City of London. And here we find that amongst the 9,535 deaths in the year 1632, some of them, and this makes interesting reading, I'm not going to go into it, were quite bizarre, but we're interested in the 1,797, which were due to consumption or tuberculosis. So in 1632, TB was causing 19% of all deaths. Now, fast forward to the modern era, it's not quite so bad because we have advances in tuberculosis care. But before COVID, and now since COVID, tuberculosis was the single pathogen that killed the most people on the planet. In 2023, there were 1.25 million TB deaths in that year. So the #1 infectious disease killer since the COVID pandemic. Of course, not everyone dies of disease. In that same year, there were 10.8 million TB cases, with a global incidence of 134 per 100,000. But you'll see that TB incidence is not homogeneous across the planet. And the tuberculosis, new cases of tuberculosis, are concentrated in the countries of sub-Saharan Africa, Southeast Asia, and to a lesser extent, South America. And I'd like you to remember this graph because we'll refer to it again when we come to BCG vaccination. So you'd think that with the improvements in TB diagnostics and TB treatments, that we'd be doing better in our fight for TB control. And on the top there, you can see that since 2010, that's TB cases on the left, TB deaths on the right. In fact, the incidence of TB has been coming down very slowly. You see that blip on the right-hand side. That blip is the COVID pandemic, which undid many years of our gains. And in fact, most of the advances in decelerating or decreasing the incidence of tuberculosis are driven by improvements in HIV care and treatment of HIV-associated tuberculosis. And the same applies for the decrease in deaths due to TB, which is shown on the right-hand side there. A lot of that driven by improvements in care for people with HIV. If you look at the milestones, our progress against the milestones which we've given ourselves, this is the NTB strategy, and it was proposed that by 2025, relative to 2015, we would achieve a 50% reduction in TB incidence rate, and we're at 8.3%. It was proposed that by 2025, we would achieve a 75% reduction in the number of TB deaths, and we're at a 23% reduction. So we're not doing very well. And the fundamental reason that we are not doing very well is that diagnostics and new diagnostics and new and better treatments are absolutely important for people who have TB, but they have had limited impact on TB control. And for TB control, we will need a new vaccine. But you might say, well, we have a vaccine already. We have BCG. I got it. Maybe you got it. Your children got it. And BCG has been around for a long time. So Bacille Calmette-Guerin vaccine was invented by a physician, Albert Calmette, and Camille Guerin, who was a veterinarian working in the Pasteur Institute in France in the late 1800s. And they cultured a virulent strain of M. bovis from a cow with mastitis, and cultured it again and again and again, and tested this strain in a variety of animal models in which it appeared to lose virulence. And it was only in 1921 that, and this is now decades of work, that they became confident enough to use this strain in a human. And there's this wonderful description of the first use of BCG given orally to a newborn infant who, as they say, was doomed to infection with tubercle bacilli because he'd have to live with his contagious grandmother. And in those days, remember, there was no preventive therapy, there was no treatment for tuberculosis. And so the chances of getting of an infant with household exposure getting TB meningitis or disseminated TB and dying from those conditions was very high. So this infant received 3 doses of oral BCG with no ill effects and remained well, despite being brought up in a bacilliferous milieu. And I've been looking for opportunities to use that term in casual conversation, but they never seem to come up. So this was the first use of BCG in humans. And over the next decade, there was an accumulation of these sort of random, uncontrolled experiments, which seemed to be successful. But it was only towards the mid-1900s that we see clinical trials of BCG that we would recognize today. And here is one such. So this is a randomized controlled trial of BCG vaccination in infants in the south side of Chicago. And in those days, this would be, I think, the 1930s, 1940s. The south side of Chicago was a very rough and ready place, socioeconomically deprived, and with a lot of tuberculosis in the urban center. And you'll see the controls above that horizontal line, the BCG vaccinated infants beneath. And there are three things I want to bring to your attention. Number one is that, as we would expect, most of the cases of tuberculosis occur in children who are younger than 6 years of age. And that's an observation that holds true today. The second is that there are far fewer cases amongst the BCG vaccinated children below the line compared to the controls. And then the last thing is that remarkably, amongst those children under 2, there are in fact no cases of tuberculosis amongst BCG vaccinated children. So this is some of the earliest evidence in a randomized controlled trial that BCG provides convincing protection against tuberculosis in childhood. And over time, there were many such trials in infants and in older children. And then if we put all those trials together in a systematic review here of trials and case control studies and other cohorts, we see that the efficacy of newborn BCG is approximately 74% in clinical trials, and with 74% efficacy, or 64% efficacy rather, against TB deaths. So fairly convincing evidence that BCG benefits young children when given in infancy. And so that's the reason that we've been using BCG for the last 100 years. And is it not amazing that when BCG was introduced, a tool that we are still using today, this vehicle was the most modern piece of machinery on the road? So we are using the Model T Ford of TB vaccines. And it's also for this reason that the use of BCG is very widespread across the planet. So in pale blue on your screen, those are all the countries in which BCG is used universally for infant vaccination. But you'll remember that in the top right-hand corner, that graph showing countries where TB incidence is high, and that those are remarkably similar countries. So what is happening there? So we vaccinate everyone against, or all infants against tuberculosis, yet TB incidence remains stubbornly high. How do we explain that? Well, the answer is not that BCG does not work, because BCG does work. It is highly efficacious. The answer is that the protection offered by BCG is not durable. And in a number of clinical trials, with one or two notable exceptions, but in most clinical trials of infant BCG vaccination, the protection offered wanes by about 10 years after vaccination, which means that by the time these children get to adolescence and into adulthood, they are no longer protected. And why is that important? It's because adolescents and adults have cavitary tuberculosis, which is the phenotype of disease that drives transmission. And so we have an ongoing tuberculosis epidemic. So we need a new vaccine, a new and different vaccine. And the WHO has kindly given us some guidelines of what that vaccine should look like. And there are two guidelines, one for infants and one for adolescents and adults. And here's the one for infants. And this is strategic priority number 2. Why number 2? Because, as I mentioned, control of the epidemic will need a vaccine for adults. And in fact, vaccinating adults against tuberculosis will reduce the rate of TB in children more than a vaccine for infants would do. If we had a new vaccine for infants, what should it maybe look like? Well, it should do all the things that BCG now does, but maybe better. relative to placebo. Not that we'd ever do this trial. Such a vaccine should have 80% or greater efficacy against disease, or it should have superior efficacy compared to BCG. But notably, it should be safer than BCG. It should be safe in babies with HIV, and it should be ideally be less reactogenic. And for those of you who have children, you'll appreciate that BCG is a highly reactogenic vaccine. What about the new vaccine for adolescents and adults? So WHO proposes, as I've stated, that the primary reason for such a vaccine should be to prevent active pulmonary TB in adolescents and adults. WHO proposes that vaccine should be at least 50% or greater efficacious, and it should protect people who've been exposed as well as those who have not yet been exposed to tuberculosis. And we'll get back to that point because that's important. It should protect people in different countries and latitudes, and it should be safe in various populations, including people with HIV. For both of those recommendations, ideally, we would like durable protection for more than 10 years without needing a booster. It would be great if we could identify something that we could measure in blood that would tell us whether a person is protected or not. That's a vaccine-induced immune correlate of protection. That's a long story in and of itself. But the schedule is also important. If you think back to the number of South Africans who received their second dose of a vaccine against SARS-CoV-2, WHO proposes that the maximum dose for a new TB vaccine should be 3. Ideally, it should be 2 or even 1. Durability we've talked about, and then such a vaccine should be able to be co-administered with the other childhood vaccines. So, you may ask, why don't we have this TB vaccine already? We heard from the last talk that there was a lot of money poured into developing a SARS-CoV-2 vaccine, and we have lots. And indeed, so Operation Warp Speed. So SARS-CoV-2 was sequenced in February of 2020. By December of that year, we had emergency use FDA approval of the Pfizer-BioNTech mRNA vaccine. Why can't we do that for tuberculosis? And the reasons are manifold. Part of the reason is that there were pre-existing lipid nanoparticle mRNA vaccine platforms that had been developed for influenza. The biggest reason, or one of the biggest reasons, was that COVID-19 was rightly perceived as a major imminent global threat, and particularly a threat to the countries of the global north that poured vast amounts of funding into SARS-CoV-2 vaccine R&D in the form of government guarantees and pre-purchase agreements prior to efficacy trial data being available. And then the last important point, and you saw it on a Kaplan-Meier graph that I think Penny showed, is that these vaccines were trialed under pandemic COVID-19 conditions in which there were very many cases during the periods when the trials were conducted. And this is why, when I last checked, and I don't think this has been updated, in 2023 there were 183 COVID-19 vaccines in development, 50 COVID-19 vaccines in phase 3 alone. And through more than 40 efficacy trials, more than 400,000 volunteers, we had 12 WHO approved COVID-19 vaccines within 3 years. So compared to COVID-19, how are we doing in tuberculosis? Well, here's how we're doing. Well, Well, I can tell you right now, we don't have 183 tuberculosis vaccines in the pipeline. We have 16, and that's if we count BCG. We do have a pleasing diversity of different vaccine platforms. So you'll see there in the orange, we have viral vectored vaccines. In red are killed or inactivated mycobacterial vaccines. And darker orange, live attenuated mycobacterial vaccines. We have protein adjuvants in green, and the new kids on the block are mRNA vaccines that have recently entered clinical trials in humans. So the pipeline is small. There are relatively few candidates. Another persistent problem has been that there are relatively few antigens. So ESAC6, antigen 85 are common to many of these candidates. So not much in the way of antigen diversity. But if you look at the pipeline, you will see that it is strangely shaped. It's in fact the mirror image of how we would expect a pipeline to look. We would expect a pipeline to have lots of candidates in phase 1 and 2 and very few in phase 2 and 3. But in fact, the TB vaccine pipeline is the other way around. And I'm going to talk to why that is. Effectively, where that leaves us now is that we have a number of candidates either soon to report or in the coming years will report efficacy data or currently reporting efficacy data. But Plan B and Plan C might be difficult in that there are not many candidates coming through phase 1 and phase 2. So why does the pipeline look like this? Well, it comes back to stakeholder sentiment, I suppose, and the roller coaster that has been much of my career of TB vaccine efficacy trial results. And some roller coasters seem to go down for more than they go up, certainly some years. But this story starts in 2013, when we published the results of the first infant efficacy trial of a new TB vaccine in 50 years, since in fact BCG. This was a trial of a vaccine called MVA-85A, and this was given as a booster injection to infants who'd previously been vaccinated with BCG. And these infants were then, almost 3,000 infants were followed for a median of 2 years for TB disease with a very strict primary endpoint definition. And you can see from the Kaplan-Meier graph there that there was no efficacy demonstrated, and that MVA-85A boost vaccination offered no additional protection to these infants. And the reaction was one of wailing and gnashing of teeth. Here's just one of many headlines along a similar theme: tuberculosis vaccine hopes dashed. And in retrospect, how naive we all were that we expected the very first efficacy trial of a new TB vaccine to be successful. We didn't expect an iterative process of getting closer and closer to our goal. Well, clearly not. But it's these results that put back stakeholder and funder sentiment for some 5 years and are responsible for the delayed introduction of new vaccines into phase 1 and phase 2 that we are still seeing today. Luckily, we didn't have to wait too long for the good news to start coming in. So in 2018, we got results, or 2017 and 2018, we got the results of a large efficacy trial of a protein adjuvant vaccine called M72, adjuvanted with AS01. It was tested in TB sensitized people, so IGRA positive people, more than 3,500 adults in 3 countries in sub-Saharan Africa, who were followed for 3 years for microbiologically confirmed TB. And the results were firstly positive and strikingly so. And you'll see towards the tail end of that Kaplan-Meier that very little evidence of waning of efficacy over those entire 3 years, a shade under, if we round it off, it's 50% vaccine efficacy in Ig-positive people, which meets, if you remember, the WHO criteria, preferred product characteristics. And the results of this trial completely changed sentiment in our field, and we'll talk to some of the new trials that are planned. But first, when I speak of sentiment, so sentiment is important. In the interim, we have then dealt with a number of trials that have just not worked, and a couple of ideas have been tested, including, and you might have been thinking to yourselves, well, if BCG given in infancy wanes, well, why not give it again? So we did this some years ago. We revaccinated Ig-negative adolescents to see if we could prevent infection. And indeed, there was 45% efficacy against sustained infection over a period of 6 months. And we hoped that this would trigger a prevention of disease trial of this strategy. But funders wanted more evidence, so we repeated that trial in a bigger population, in a wider age range, and at more sites. And we convincingly showed that, in fact, BCG revaccination does not prevent sustained Ig conversion in adolescents. And ultimately, that's what you want from a trial. You want a clear answer, and I think again that the data from this trial provided a clear answer. And in recent years, we've had a number of such trials with a sort of experimental medicine flavor or proof of concept flavor. This is a protein adjuvant vaccine called H56, which was tested for prevention of recurrent TB in TB patients who were deemed cured at the end of treatment. And you'll see on the left-hand side there that there was no evidence of efficacy against recurrent tuberculosis. And on the right-hand side there, if we drill down, although the numbers are small, if we drill down into the patients who relapsed, in fact, vaccine had was associated with an adverse effect, in fact, a higher rate of TB relapse. So H56 is no longer in development. And then finally, a recombinant BCG called VPM1002, which has recently been tested in a very large trial of 13,000 individuals in India for prevention of disease amongst household contacts. Those results are still officially awaited, but the WHO chief scientist is quoted in the press and helpfully tells us that overall there's not much difference between the arms. So we'll take it that, in fact, efficacy against the primary endpoint was not achieved. So the curious thing about all these results is that the field as a whole has gone, okay, but what's next? And what's next are two pivotal efficacy trials in adolescents and adults that are likely to, we hope, write those last few pages in that final chapter of the story of TB vaccine development. Because as our chief scientist from WHO, Jeremy Farrar, commented last year, funders like to fund things that work. And there are two vaccine candidates that seem like they might work. And the first I've told you about already is M72, which is that vaccine that showed efficacy in a phase 2b trial in 3 countries in sub-Saharan Africa. And this vaccine, it took 7 years, but this vaccine is now funded to the tune of more than $500 million to fund a phase 3 licensure trial amongst 20,000 people at 54 sites in sub-Saharan Africa and Asia, who are predominantly, again, Ig positive, so TB sensitized, and living without HIV. and we'll talk to the implications of those findings in a moment, and results expected in 2028. I won't go into details, but the second vaccine that's causing a certain amount of excitement is a vaccine called MTB-VAC, which is a live attenuated tuberculosis, which has 2 balanced deletions. And this is a vaccine, because it's a live attenuated tuberculosis, it has all the antigens that M. tuberculosis has, that M. bovis has not. And it's also got all the antigens that fell out of M. bovis when BCG was made. So it has a considerable antigenic diversity, which may or may not be an advantage. And that vaccine has been shown to be similarly reactogenic and, in fact, more immunogenic than BCG at the same dose, and has recently entered a phase 2b trial in over 4,000 people, with results expected in 2029. I'll skip that slide. So what do we need to progress TB vaccine development? Well, very simply, a lot of the issues can be fixed with funding for more candidates, more preclinical work, more clinical work, and more efficacy trials. And TB vaccine development is woefully underfunded. But the investment case is very strong, in that a vaccine with only 50% efficacy could avert tens of millions of new cases, more than 8 million deaths, and billions of dollars of treatment costs. So it is a travesty that TB vaccine development is funded largely by the same club of philanthropic institutions. And I'll leave you just in these last 2 slides with 2 ideas of things that are missing from the plan. And you might have got a sense that in fact children and TB unsensitized people, so IGRA negative people, are missing in action from TB vaccine efficacy trials. These trials are not powered to show efficacy in these groups simply because too few TB cases would accrue. So trials are not practical. So we will not have data from IGRA negative people. And that's important because IGRA negative people respond to vaccination very differently from IGRA positive people. And quite simply, we will not have data to show that children or IGRA negative people would benefit from a TB vaccine. So the plan, as we understand it, is for these new products to be licensed based on efficacy in IGRA positive people and safety in IGRA negative people. So when we think ahead to licensure and implementation, we will have efficacy data in IGRA positive people aged 15 to 44. We will have safety and immunogenicity in some other subgroups. We will have no data from children. We will have no data from the elderly. The elderly, you're hearing it from me, is anyone over than 45 as far as the TB vaccine development community goes. We will not have data from people living with HIV who are not established on antiretroviral treatment. People who have previous TB, women who are pregnant or lactating, or from specific risk groups. So these obviously have implications for potential label restrictions and for targeting of rollout. So in summary then, I hope I've left you with the idea that there's a great need for a new vaccine, that the pipeline is small, there's few early phase candidates, but we have 2 very promising candidates in efficacy trials. And we will have efficacy data, although from a limited subset of the population by 2028 to 2029. But we will not know for the purposes of rollout if the entire population will be protected. So that's the final chapter of TB vaccine development that is yet to be written. And I'll just leave you with thanks to all the investigators that have contributed to these studies and to our colleagues at Stellenbosch and elsewhere, and particularly to the study participants and their communities who live in places where tuberculosis is such a burden, but without whom we could not do these studies. Thank you.
So, we can have one burning question, and so there's two. Which one?
Thanks for a fantastic talk. Maybe I missed it, but with the M72 AS01 construct, I didn't read the paper, but was there protection against pulmonary TB?
Yes. Okay. So the endpoint in the M72 trial was symptomatic, microbiologically confirmed pulmonary TB. So no trials to my knowledge have used extrapulmonary TB, for example, as an endpoint, and that's quite simply due to the fact that it's numbers, it's logistics. The study population would just be too large.
Thanks. So